All right, ladies and gentlemen, welcome back. So today we're going to talk about blood vessels. And so when we look at blood vessels, let's just talk about some basic characteristics. First off, when we look at a typical system like ours, we talk about three main types of blood vessels: your arteries, your capillaries, and your veins. And keep in mind that these are distinguished not really because of oxygen content, but really by the direction of which that blood is flowing through those vessels. Are they going towards the heart, away from the heart, or are they sort of a connection in between? So your arteries are carrying blood away from your heart. Most of the time when we're talking about this, you think about the fact that they have high oxygen content, and that's true when you talk about your coronary circulation and your systemic circulation. But remember, when we're talking about your pulmonary arteries, these guys don't have high oxygen content. Instead, it's low, because we're pumping that blood to the lungs to pick up that oxygen. So that's sort of where that caveat comes in. So be careful. So anything that carries blood away from the heart, that's a pretty solid definition for an artery. Um, your bigger arteries then feed your arterioles, which are smaller, or still feeding away from the heart. And those arterioles then feed your capillary beds. And your capillary beds are the actual site of chemical exchange between your blood and your tissue fluid, your body cells. So the capillaries are relatively thin to facilitate that diffusion that can only happen over real short distances. And at one end of the capillary bed, it's going to be high oxygen content. That's the end that the arterioles are feeding. We're going to push that oxygen, for example, those nutrients out into your tissue fluid. And then on the venule end, which will eventually feed into your veins to go back to your heart, the vein end, the venule end of your capillary beds is oxygen poor, and that's going to then feed back to your major veins if we're talking your systemic circuit. Um, your veins, then remember, are the ones that carry blood back to your heart. Again, be careful with that caveat because when we talk about your pulmonary circuit, those oxygen contents are flipped, right? So veins in your pulmonary circuit have high oxygen because they're carrying that stuff back from the lungs. So just be careful with that oxygen content when you're talking about it. Um, but if we look, the flow of blood is always in one direction, or at least it's supposed to be. So you go from your ventricle in your heart to your major arteries, whether it's pulmonary or your aorta. Those will then feed into smaller arteries called arterioles, which feed into your capillary bed, which is where that exchange of materials happens. That then feeds into your venules, which are small veins, which feed into your major veins, things like your superior and inferior vena cava, or your pulmonary veins, which go back to your heart, specifically to the atria. So when we look at the basic structure of these arteries and these veins and these capillary beds, it very much is connected to the function that they play. So when we look at your capillaries, for example, all right, there really isn't much to them. They've got some basic endothelial layers in the center, which also line your arteries and your veins as well. And then they have a real small single cell layer called your basal lamina, and that's it. We need these to be incredibly thin vessels because we need to be able to feed out to your interstitial fluid, your tissue cells. And remember that diffusion of gases and nutrients and materials isn't really good over big distances. So the thinner we can make this and still serve its function, the better. Your arteries and veins also have those endothelial layers, but those are then surrounded by smooth muscle cell and then by connective tissue, because again, these aren't really the sites for that exchange of material. Instead, I just need to get that blood, that fluid, from one place to another. If you look, your arteries have a incredibly uh, thicker layer of smooth muscle and connective tissue than compared to your veins, and that's on purpose. We need to be able to handle the high pressure of your blood pushing that blood through those arteries to your body. So we need to accommodate that much higher blood pressure that you're going to see in those arteries, which is why nicking something like your femoral artery, you'll actually see it spurt, right? Every time your heart pumps, you are going to increase that pressure, and if it's got nowhere to go, it's going to come spurting out of your body. Your veins, on the other hand, have much lower pressure because we've lost a lot of that blood pressure here in your capillaries. All right, so heart pumps, lots of pressure here, decent amount of pressure here, but in order to slow the blood down so that that exchange of material can happen effectively, we've got to lower the pressure, slow everything down, which means that that blood is moving considerably slower and at much lower pressures when we get to your venules and then back into your veins, which then causes its own problems. So I don't need as high of a muscular wall to maintain pressure, but I also need to keep the blood moving in a single direction. And without that pressure, think of trying to push something from your foot back up to your heart if you're standing up. It becomes a lot more problematic because there's no pressure to keep it moving. And so you actually have these valves in your veins to keep that blood moving in one direction, very similar to valves in your heart. So if we were to look at these guys under a microscope, you can see the big difference here. So here's that lumina, or that opening where your blood would flow. And if you look, it's much tighter, much thicker muscular wall here in your arteries. They maintain their shape much more. Where if I look at a vein, much smoother, much thinner layer of muscle. And as a result, they tend to be a little more squishable, if that even makes sense. You don't have as tight of an opening that gets maintained. It gets a little more wavy as it moves around because you don't have that extra muscle giving it that super rigidity that you need in your arteries. So I mentioned this idea of blood flow velocity, right? How quickly is it moving? And that's tied to the need for that thick muscular wall in your arteries and not so much in your veins. And this is exactly what we're looking at. So both area that they cover versus the velocity or the speed that that blood is moving through them. And then that's directly related to the pressure itself. So your capillary beds have to have the lowest velocity and as a result end up losing pressure as a result in order to maintain that flow of nutrients from those capillaries out into your tissue fluid and vice versa. So we need to slow everything down as a result. So that high resistance is a result of just a lot of area as opposed to speed or pressure. And that increase in area and drop in speed and pressure is necessary for all of that exchange. So your blood pressure is just that. It is the pressure that's exerted outward against the walls of the vessels, and then what sort of pressure can they push back based on the elasticity of the materials that they're made of. So blood that flows directly from your heart is getting forced with a lot of pressure out into those major arteries. And so you'll have much higher artery pressure here. And every time your heart pumps, that pressure increases. All right. So your diastolic pressure will drop, right? That's when your heart's relaxing, and then increase when you get those pumps in your systolic pressure. So every time your ventricle relaxes, your blood pressure drops a little bit. Every time that it pumps and goes into systole, you get spikes. And so just like anything else, when we talk concentration gradients, or in this case, pressure gradients, your blood is going to want to continue to flow from where it's high to where it's low. It's going to constantly be seeking out lower pressure. And so that's going to keep that blood moving through your arteries until we get to those capillary beds, things slow down. And then we get into your veins where your blood pressure pretty much drops off completely. And that's where those valves help to keep things moving in the right direction. So again, I mentioned this term systolic versus diastolic pressure as a result of your systole versus diastole of your ventricles. So anytime you've got that systolic pressure, right, that's when your ventricles are in systole, that's the highest pressure that we're going to measure in your arteries. Diastolic pressure is the lowest that we'll measure in your arteries. So it's all about arterial pressure. And that constant rhythmic up and down, that bulging of those walls, is your pulse, is your heartbeat, right? And again, I mentioned to you before, if you trace down from your ear along your jawline, just below your jaw, you should be able to feel that change in pressure as your pulse when you find your carotid arteries. And same thing in your wrist, if you can find out that one as well, you'll feel that constant spike and drop in pressure that we typically associate with your heart's pulse. So your blood pressure is really important to keeping that blood flowing. That does mean that there is a homeostatic mechanism in place in order to ensure that that is the case. And so let's take a look at that pressure regulation. So it is a negative feedback loop, all right, meaning if you get imbalanced, your body's going to try to do the opposite to you to bring it back. So what part of that connection is not only your heart rate, but then also your dilation of your blood vessels. So if we get a situation where you've got a rise in your blood pressure, you have receptors in places like your carotid arteries to look for that, to monitor that. And as that blood pressure is changing, it's going to send signals to your brain. So if we have a rise in that blood pressure, that's going to go, nope, not supposed to do that. And so signals are going to come back from your brain, both to your heart to try and decrease your heart rate, but also to the major blood vessels in your body, mostly your arteries. And what we're going to cause this to happen is to vasodilate, meaning we're going to increase the diameter, increase this space here for that blood to flow. What are we doing? We're really causing the muscles in that artery to relax. All right, and so when they relax, they allow that diameter to open up. That should then pull you back into your normal blood pressure range, balance you out, return you to homeostasis. If we see a decrease in your blood pressure, all right, same receptors send signals to your brain. This time we do the opposite. We increase your heart rate, try to increase your cardiac output, as well as increase what we call your peripheral resistance by causing your blood vessels, your major arteries, to now constrict. We contract, vasocontract the smooth muscle in your artery walls, decrease the opening for which that blood can flow, which increases the pressure and should hopefully pull you back into homeostasis. And we can measure this, right? So if you've ever had your blood pressure taken, then you have in fact felt what this feels like. So generally we measure a major artery in your arm, right? Usually right kind of in the crux of your arm here, and at approximately the same height as your heart. So they'll put the blood pressure cuff on you, right? They'll squeeze up the pressure, tighten that major artery in your arm. Nurse or doctor will sit there with the stethoscope listening for the heartbeat basically to stop, right? We cut off the blood flow, and then they'll use that little knob to slowly release the pressure. And as they do that, they're listening for how many beats come through as a way to measure both your systolic and your diastolic pressure. So average healthy adult, all right, usually we say somewhere on the order of 120 systolic over 70 diastolic. That number sometimes changes a little bit depending on recommendations, but average we're saying somewhere around 120 over 70, 120 over 80 is a pretty healthy blood pressure for most average functional adults. All right, let's dive back into your capillaries a little bit. So we're looking at that need for exchange, right? So only about 5 to 10% of your body's blood coming from your arteries is actually flowing through your capillaries at any given point in time, right? Once we get to those arterioles and we get into those capillary beds, we're going to slow down the velocity, slow down their speed, um, and really decrease the pressure as a result as well. So your capillaries are going to surround all sorts of tissue fluid, your major organs, all right, as a way to get them the oxygen and nutrients that they need and get rid of the waste products as well. Okay. And it does this really by this basic mechanism here. So you'll have a major thoroughfare channel, which connects basically your arteriole on one end to a venule on the other. And then you've got these branched out components to the bed itself that really are going to do the feeding. And you've got these capillary sphincters here on the arterial side of your capillary bed um, that either allow the blood to go into those branches or keeps it within the thoroughfare channel and not really feeding the tissue itself. And the opening and closing of those sphincters is done by contraction of muscle cells, all right, and is once again driven by your nervous system and those feedback mechanisms. So if I really need my body to be in fight or flight, well, then I'm not really worried about digesting my food and sending tons of nutrients and oxygen to places like my intestines or my stomach. So we may shut off the sphincters to their capillary beds in order to get a lot more muscle uh, blood flow instead, right? So hormones, nerve impulses, other sort of chemicals can all control whether or not those sphincters are open and whether or not we're allowing the perfusion of blood into those capillary beds to feed that major organ or set of tissues at that particular point in time. And it comes back to homeostatic mechanisms, which is a good evolutionary component to this. The fact that we have evolved the ability to shut off blood supply to certain things when I don't need them and be able to reroute that blood um, when necessary. So your capillaries, right, are there to be that major connection between the plasma, the fluid of your blood itself, and the interstitial fluid and materials out there. So again, the speed of the distribution of nutrients, hormones, dissolved gases, all of that is facilitated by the fact that these capillary beds are really, really thin. Okay. They'll also flush antigens to your lymphatic tissue, so there's a connection here to your lymphatic system as well as to your immune system, aids in the movement of proteins in places where we need them, keeps water flow and water balance hopefully in play and in check through osmosis. All of that is happening here at those capillary beds. So again, if I look here, right, here's just another way to look at this. Your osmotic pressure should maintain and be constant, right? Keep the fluid both inside and outside at an isotonic state. Okay, if you get a lot of leaking out of your capillaries of water, there's something going on if you're not returning that back to circulation appropriately. If we look at the arterial end of your capillaries, right, that's where the pressure is going to be highest. That's going to drop off until we get to the venule end because that high blood pressure is going to be pushing all of that material out, so outward flow, right? We get to the venule end and then we're going to get inward flow based on the concentrations of nutrients and oxygen and water and the whole 9 yards that's going on there. We'll get a little more into that when we get to the second part of chapter 42 notes. Most of your blood proteins, your blood cells, those are all too large to flow through. So it's all about those small molecules, water, and that sort of thing that are going in and out here. It's not the red blood cells, generally not your white blood cells unless there's a reason for it, and then some of those cells are capable of squeezing through. Platelets shouldn't be going through unless there's a problem either. So most of your major cells will stay within circulation and not go out into your tissue fluid unless there's a reason for it. I mentioned to you a moment ago, right, there is a connection to your lymphatic system, which also connects to your immune system. So anytime we are leaking fluid out into your body cavity, into your tissues, that is then considered lymph, right? And this fluid gets sucked up by your lymphatic vessels, often goes through those lymph nodes as a way to sort of check, and that's the connection back to your immune system. And it often gets dumped back here into your subclavian vein, which empties into your vena cava and drains back into your heart. Okay. Just like your veins, however, there isn't a whole lot of pressure in these lymphatic ducts, these lymphatic vessels, and so they also have valves, just like those veins do, as a way to keep that fluid moving so that it gets back into circulation rather than getting stuck somewhere in your body and causing that particular body part to swell up.